You haven't heard the talk yet, so clap only at the end. Thank you so much for inviting me. This is an honor. I'm a very big fan of the CRESS team and the work that's being done, and very happy to be here in Paris. I apologize. I'm also one of the two speakers that does not speak French. So that's my fault. But hopefully it'll still be interesting in English, and I'll try to go as smoothly and clearly as possible. So today I want to talk about some work that I've been doing thinking about the use of real-world evidence and observational research, in particular with a focus on informing regulatory and health policy decision-making. So I'll start with something that we likely all know and think about, just setting the framework for why we're thinking about this. So randomized controlled trials, they're often considered the gold standard. We saw in earlier talks the evidence-based pyramid. They're used to inform our understanding of the efficacy, effectiveness, and safety of medications and therapeutics. Because of our randomization, we have minimized confounding and balance between groups. This can minimize systematic differences in treatment allocation. And because of these reasons, these RCTs, or randomized controlled trials, are often used to inform the decisions made by regulatory bodies, patients, physicians, clinical guidelines, etc. But despite these strengths, and in response to one of the talks, yes, we still do need randomized controlled trials, but my talk wouldn't be as interesting if I didn't set up some limitations. So there's also limitations when it comes to randomized controlled trials. So if you think about how randomized controlled trials are designed, they have strict inclusion and exclusion criteria that minimize the types of populations that are identified. And because of these inclusion and exclusion criteria, we often lack generalizability beyond the sample that we're studying. And this can have impacts if we're thinking about things in terms of race, ethnicity. Older adults are often excluded from randomized controlled trials. So because of this, RCTs can often have limited generalizability. Another limitation, of course, is cost. In a previous talk, there was a talk of pivotal trials, or RCTs, that are used by the US Food and Drug Administration to guide the approval of new therapeutics. Evidence suggests that it's approximately $20 million for a clinical pivotal trial. Cost, operational challenges. And by operational challenges and operational complexity, I mean that it's often difficult to recruit patients into trials, especially if we're thinking about rare diseases and groups in oncology that may have small samples that would make it difficult to conduct larger randomized controlled trials. And along with difficulty recruiting patients, we also have the issue of retaining patients in our RCTs. So what happens when patients no longer participate after they're recruited? And then we also have this issue of evidentiary gaps. And what I mean here is that RCTs can't be used to study every single question. There are certain comparators that will never be compared. Either it's not ethical or there's no financial incentive to conduct a randomized controlled trial comparing two interventions. Another issue is that they may have a short duration, which would mean that you couldn't study the long-term efficacy, effectiveness, or safety outcomes, or they would be measuring outcomes that don't tell you how people feel, survive, and function in the long term. They're designed particularly to be shorter and measure intermediate endpoints. So because of these limitations, there's been growing interest in this idea of, all right, causal inference from observational data. And I guess this is not a new idea at all, but there's renewed interest in the kind of intense application and thought process going into how we design our observational research. And the overarching question and item that I'm going to be thinking about and talking about is, can we use real-world data, and I'll define that, and observational methods to address the same clinical questions as studied in randomized controlled trials while minimizing bias and confounding as much as possible. And the reason that this has become a hot topic or a buzzword is that there's growing interest from the regulatory perspective. You can see here's, it might be easy or hard to see. These are guidance documents from regulators. One is from the US Food and Drug Administration about how to use real-world evidence and observational research in drug development. One is from the EMA about real-world evidence and the framework to support approvals. So there's a push to use real-world evidence and observational research methods to inform how drugs are approved. So what exactly do I mean by real-world data? Essentially, we're talking about observational data or data that is collected outside of a randomized controlled or clinical trial. So these could be electronic health records, patient-generated data through devices that may be collecting information on health status, claims and billing data, so administrative claims data, and disease registries. So in this buzzword umbrella of real-world evidence, you can think of there's many different ways that we can collect information on patients outside of randomized controlled trials. And then we have another buzzword, target trial emulation. And this is really what I'm going to be talking about here. Although there are many types of observational studies, there's been growing interest in using or applying the target trial emulation framework. And this is a framework designed to limit the bias in observational studies by attempting to recreate a randomized control trial or emulate or copy a randomized control trial using real-world data. So the goal is to identify a target of interest or a target trial of interest and try to design an observational study that looks like it as closely as possible. So how does one emulate or copy a randomized controlled trial? How does this actually work? And this is not a long talk. I could spend, a whole day on most all of these issues, so this is going to be rapid. And I'm welcoming questions at the end, but I should point everyone into the directed readings that are kind of cited at the bottom. But first, our goal is to identify a hypothetical trial, so something that we want to study but may not be conducted as a real randomized controlled trial, or an actual target trial that's being conducted. And here you can see you can look on different places where randomized controlled trials are being conducted, clinicaltrials.gov or international clinical trial registries, and you can see trials, identify your target, or design one that is a hypothetical true trial that cannot be conducted. After we've done that, we need to identify our data source. And I said real-world data source, so at least when we're thinking about target trial emulation from US perspective, we're often using two types of data, administrative claims data or electronic health record data. And by administrative claims data, I mean these large data sets that are generated for billing purposes and reimbursement, and they capture information about health services. And this is great because they can capture information about patients across health systems, but they're not actually designed to study health in a way that we ideally want to. They're designed to support payment. So we have information in this source, but it's potentially limited in what we can do with it. And then we have our electronic health records, and this is more detailed patient health information. We can have lab results, imaging, clinician notes, and this is really used to inform clinical decision making. Now, this is great because we have improved granularity when it comes to the information that we have, but the issue here is that the data may not be as easy to work with, or it may not capture information outside of a certain hospital that's using this information. And we often have unstructured data, and what this means is that it's not actually captured in the data set in a way that is easy for us to analyze outcomes or patient characteristics. If we're dealing with notes describing a patient, how are we going to translate that into something that we can use in our observational research? So you can already see how I've talked about limitations of randomized controlled trials, but how limitations here are starting to introduce themselves, at least as we're thinking about our data sources. So once we've identified our target trial of interest, our data source of interest, we think about the key components of a trial. And this is where you write out your hypothetical or true trial characteristics. And the goal here is to design your observational study in a way that matches as closely as possible a real randomized controlled trial. So we think of all these characteristics that we normally think about in a randomized controlled trial. The eligibility criteria. And the goal here is to have a detailed inclusion-exclusion criteria that matches as closely as possible to what a real trial would look like. Again, you're designing your observational study with the criteria that you would design your randomized trial. Treatment strategies is our next point. And what we want here is to think about, okay, what can we feasibly study using observational data? In reality, we're going to be wanting to establish trials that are pragmatic in a sense. So we're comparing treatment strategies or prescriptions of medications. We're not actually comparing true randomization of an intervention to participants. So we have to think about that when we're targeting our trial of interest. Then we want to think about our treatment assignment. And this is where things get, well, I shouldn't say this is where things are complicated all along, but this is where things get particularly complicated from our epidemiological and causal inference perspective. We want to emulate randomized assignment, so we want to get as close as possible to random assignment by adjusting for confounding factors. And this kind of sounds like I'm waving away saying like, and we can adjust for confounding factors and it'll look like a randomized controlled trial. That's not necessarily what I'm saying. I'm saying this is where we're thinking about our causal inference framework. How do we actually design this in a way that it's going to look as balanced as possible between our treatment groups? And then we want to define our study start and end times. And this is a very simplistic overview of what this looks like. But in order to minimize our immortal time bias, which arises when treatment strategies are defined after startup, we want to define our use of a treatment at time zero. This is new users. We want people who are newly prescribed medications or newly receiving treatments. And then we want to follow up across different groups the outcomes that are observed, whether it's an outcome or a death or loss to follow-up if they switch out of the health plan that they're in. So the objective here is to identify new users of treatments in multiple groups, 2 groups here, like a randomized controlled trial, follow up until the event or some other outcome occurs that disqualifies them, and ensure that we have enough information in our database about these patients that we're studying, because we need baseline information on our patient participants because we need to be able to adjust for confounding. So we build in a look-back time of 6 to 12 months usually, ensuring that these people are in our database for long enough so that we can observe their baseline characteristics or their medical histories. And then we want to think about what our outcomes are. And this is also where things get tricky because we have to study outcomes that are feasible to capture in real-world data. And I described these 2 sources that are often used, administrative claims data and electronic health records. We're limited to the information that we can capture there. So studying behavioral outcomes are challenging here because they're not actually structured in these data sets. And then our causal contrasts. And when we think about causal contrast here, I'm talking about, okay, is our randomized controlled trial objective to conduct an intention to treat analysis or a per protocol analysis? Is the objective to look at adherence to medication and the outcome, or look at patients based upon what they were randomized to? And here in this target trial emulation framework, the closest parallel to intention to treat is the comparison of initiators. So you're comparing people who are hypothetically initiating treatment and then following up to see what happens. So then we formulate our analysis plan, and the goal for the analysis plan is to mirror the randomized controlled trial as closely as possible. We want to conduct a Cox proportional hazard model if that's what the randomized controlled trial says they're going to do in their study protocol. But our objective here in an emulation is to make sure that the patients are equally likely to receive the interventions in the real world. And because of this, we have to actually balance our confounding factors between our 2 treatment groups. And there are many different ways to do this. Again, like I said, this could be a whole separate talk, but you can imagine using something like propensity score matching, where you look at the likelihood that someone is prescribed a medication based upon all of their baseline characteristics, and then match people based upon these propensity scores in the treatment arms. And then we conduct our emulation. So we identify our new treatment users in the real-world data. We ensure a look-back period for our baseline variables. We implement the inclusion-exclusion criteria in the real-world sample. We ensure, easier said than done, that our confounding variables are balanced, and then we conduct our analyses. And I hope this is right, because as I told you, I don't speak French, but I hope AI is better than I am. But is this easier said than done? And this is where, I know that was a long-winded background, but this is where we're going to get into the research experience. So let's explore some examples that we've conducted. So I'm going to be talking about 2 target trial emulations that I've worked on in collaboration with the US Food and Drug Administration. The objective here was to understand what the promise and potential of target trial emulation is when we're studying drugs and thinking about it from a regulatory perspective. What can we learn using these data? So the first trial that we selected, so again, Here we're looking at true trials. We identified existing trials, not hypothetical trials, and we tried to copy them as closely as possible using our data. And one of these, the PRONOUNCE trial, was a RCT, large, international, comparing two treatments for people with prostate cancer and cardiovascular disease with an outcome that could be captured in our data set of adverse cardiovascular events made up of death, myocardial infarction, and stroke. And I should say that we selected trials that hadn't been completed. We did not know their results. We selected registered trials, and we used the information on these trials to design our target trial emulation without, that's nice, but without being biased by what the results from the trial would be, which could inherently potentially impact what we're observing. And our second one was a cardiovascular trial. And this was comparing second-line glucose-lowering medications among patients with type 2 diabetes with an outcome of time to primary metabolic failure. And what's interesting here is we picked two large trials that were ongoing with different types of endpoints. One requiring more detailed information from electronic health records, HbA1c, and the other one, which we could identify using structured information in administrative claims data. So again, these were designed with these potential challenges in mind. I talked a little bit about this, but why PRONOUNCE and GRADE? Well, PRONOUNCE looks a lot like the type of trial that would be conducted after a drug gets approved to fill evidentiary gaps. So you can imagine a drug gets approved and all of a sudden there's concerns about safety. The FDA could require a pharmaceutical company to conduct a post-market study or study after the drug is approved to inform our understanding. So this looks a lot like the PRONOUNCE trial, a post-marketing study to inform safety in the long term. So, okay, could we use a target trial emulation to get the same information? And then the GRADE trial is a nice example of something that has multiple comparators and a laboratory-based outcome that introduces its own challenges. And these are the characteristics. We have to have FDA-approved drugs here with at least 3 years on the market. We wanted to make sure that we had enough information in our data set. We wanted large trials because we didn't want to compare target trial emulations with thousands of patients to clinical trials with 100 patients. That didn't seem like a fair comparison. We wanted feasible outcomes, and we wanted the profile that looked like things that could actually inform regulatory decision-making. So then just to tell you a little bit about our data set, we used a large administrative claims data set. So if you think here that the US population is over 330 million patients or participants or humans, we have claims data here for about 130 million. So this is our structured claims information. And then for some subset of those, about 83, we have additional clinical information from electronic health records. So we have a large administrative data set that is reasonably representative of the US population. So we use propensity scores in ours to emulate randomization. So looked at the likelihood that people were assigned essentially their treatment based upon their baseline characteristics. And in the PRONOUNCE trial, we matched people in the treatment groups using one-to-one nearest neighbor matching. And in the GRADE trial, based upon the characteristics, we used inverse probability treatment weighting. And again, these are all things that could go in a more detailed talk, but I don't think this is the most interesting part of it. This is kind of getting us there. So what exactly did we find when we conducted these target trial emulations? So one, eligibility criteria has a really big impact on the number of patients identified. So you can see that we started with a really large administrative data set, but as soon as we start implementing these eligibility criteria from the trial, our data set shrinks. And you can see this here that only approximately 20 to 25% of the patients treated with these medications were ultimately eligible for our emulation because we're looking for really specific characteristics that match a trial. This is hard to read, but essentially that's another figure showing what happens when we start to implement the inclusion-exclusion criteria. But despite this impact of the inclusion-exclusion criteria, our emulation, so our observational versions of the randomized controlled trials, were larger than the randomized controlled trials themselves once they were published. So the PRONOUNCE trial, when we were designing it, it said on clinicaltrials.gov this is going to be a 900-person randomized controlled trial. when they were published, turned out they had difficulties with things that I talked about with RCTs, recruitment and retention. 544 participants were included. Our emulated trial had 2,226 propensity scored matched individuals. The GRADE trial, which was a very large diabetes trial, had 5,000 participants. Our emulated trial was still larger, 7,500. So now, I'm not going to say things start to go downhill, but start to get more complicated. So many patient characteristics were not ascertainable using structured real-world data. And what I mean by this is many of the characteristics that we'd hoped to capture about individuals for our emulated trial aren't actually structured in our data set. And I listed this before when I talked about the potential limitations of these data sources. And here's another way to look at it. By ascertainable, I mean we can identify using validated codes these characteristics in our data set. Partially ascertainable, the middle column, represents things that we had to change based upon what actually is available in our observational data. So we had to make adjustments because smoking isn't defined the same way in our claims data set as it's defined in an RCT. So we had to change it. It's a close approximation, but it's not a perfect emulation of the characteristics. And then you can see here almost a third of the characteristics in the trial were not ascertainable at all. There was no direct corresponding item in the data set that we could capture. And you can see many of these are our clinical laboratory characteristics that aren't routinely available for all participants. So these are all of the baseline table 1 characteristics of the pronounced trial. 2/3 either had modifications or were not available. 1/3 we could implement using validated codes. And it's the same for the GRADE trial. 34 characteristics were included in the table 1, the standard table 1 from a randomized controlled trial. Of these, 41% needed some kind of modification, and approximately a third were not ascertainable. Again, these were characteristics that we just could not study. They weren't structured in a way that we could analyze them. And on top of this, our fourth finding is real-world populations are just inherently different from randomized controlled trials. And this may not be surprising because randomized controlled trials are controlled. It's a controlled environment with strict inclusion-exclusion criteria. They often may not represent who ends up taking the medication in the long run. So there are inherent differences. And we're going to go back here to look at, in red, all of the characteristics of these patients that were different between our emulation and the actual trial when it was published. And we did side-by-side comparisons of the observational population and the trial population. And the red ones are those with standardized mean differences of greater than 0.2. So there's some difference here in terms of all of these characteristics listed. And this is confusing to look at, but it gives you some sense of what is actually happening. In the real-world data set, I have an arrow up saying that we had an older population, which potentially is a good thing, greater generalizability to older populations. We had greater diversity in terms of race and ethnicity. But then we have some differences in terms of myocardial infarction. Our group had fewer at baseline than the trial itself, and some of these previous treatment medications, you can see cardiovascular medications, there were a lower number of patients in the real world taking these than in the trial itself. So we're already introducing the fact that we've emulated the trial as closely as possible, but they're not looking exactly the same when we've studied them. So what impact is this going to have on our outcomes? And it's the same for the GRADE trial. There's similar themes here that we have, improvements in things like age and representation, but differences in terms of baseline characteristics or how sick or not sick individuals are. And despite these differences, for our two examples, we found that emulated trial results and published trial results were still often concordant. They still gave us somewhat of the same information. So here I'm setting up, this is like the table 2 of these publications, primary findings from the first column, the emulated trial, so that's our attempt with observational data. The second, the pronounced trial when the results were available. And we looked at how much they agreed. Are they in the same direction? Are they both statistically significant at p-value less than 0.05? And are the 95% confidence intervals overlapping? And you can see here for our primary outcome, 1.18 and 1.28. basically giving us the same information. Okay, so now let's look at the secondary ones, which are the components of our primary outcome. Looks good for myocardial infarction, looks good for stroke, all-cause death or all-cause mortality, opposite direction. So what's happening there? Well, this is where we have this issue of potential residual confounding, we suspect. What potentially could explain the fact that we're getting the opposite effect for something like all-cause death? Well, could patients who are more severe in their illness be prescribed a certain medication that's preferable for that, and that could impact the number of patients who die because they're more likely to be prescribed this treatment? Well, we can't actually adjust well for this because we don't have information on cancer stage in our database. So you can see here how residual confounding is creeping in because we can't account for something that's critical for an outcome of interest. What about the GRADE trial? We would summarize this if this was a publication as they were largely concordant. There are some that agree and some that disagree, but we're seeing the direction of the magnitude of effect very similar, with some differences in terms of statistical significance and overlapping 95% confidence intervals. But what's interesting about this one is that one of the treatment arms of the randomized control trial was not included in our evaluation because we couldn't identify a large enough sample to match with. And this is because this trial was initiated over a decade ago, and treatment practices have changed so much over the last 10 years that you can see this doesn't even capture like GLP-1s or other treatment mechanisms that are used. So it's no longer reflecting our current treatment profile. And that means that something that was designed in 2012 may not be representative anymore. And the trial results were published in, I don't have it here. Oh, I do have it here in 2022. So by the time these results are available, one of the treatment arms is no longer really used in treatment practice. So what did we learn? I think I've hinted at this a little bit is, okay, target trial emulation strength, we could do this at a fraction of the cost. In a way, I wish we were paid $20 million to do this. This is not a $20 million analysis. This was also done at a fraction of the time. This is something that we can conduct in 12 to 18 months. If we had a larger team, it wouldn't take as long. Sample size, we have a larger sample size in both of these than the actual trials themselves. We had a rigorous real-world data source, even though there are limitations that are inherent to it. We followed the target trial framework, which improves our thinking about things like causal inference. And we had a larger population of older adults and greater diversity in terms of race and ethnicity, which allowed our emulations to conduct subgroup analyses that were proposed in the RCTs, but weren't actually conducted in the RCTs. So feasibility of conducting these depends upon the ability for us to capture the information that we want. We don't always have the laboratory data that allows us to understand the staging of a disease. For example, for prostate cancer, we couldn't get information for everyone on PSA or other information that we need to accurately adjust. And then there's the question of, should we expect the same results between randomized control trials and observational studies? We already talked about that these are inherently different populations. So does it make sense to say, all right, we expect the exact same effect estimate, the same level of significance, if our population profile is so different? I showed you that table with the red labeled items and the arrows going up and down. These are different populations. Maybe it doesn't make sense for us to say that they should be concordant. And I have just a little bit more, and I'll be over, I promise. So the question here is, in terms of implication, is when are target trial emulations feasible and informative? If this is our experience, what is the reality of implementing these in the real world? And the reason that this is interesting is this is a headline from, I think, 2 years ago from the Commissioner of the Food and Drug Administration that says, expect to see more real-world evidence-based regulatory decisions from the FDA. I could put up a similar quote from the EMA. I showed the guideline. There is interest in using this information and relying on it. So we conducted a series of studies that said, okay, if we try to emulate studies that are used by the FDA, how feasible is it that we could actually conduct these target trial emulations? And we took, in one example, all of the post-marketing studies for accelerated approval drugs, and essentially these are drugs that are approved faster because they target a disease that has an unmet need. There's a need to develop medications, but they're approved with limited information with the expectation that additional clinical trials will be conducted after approval. So we took those trials, we looked at their characteristics and said, could we conduct a target trial emulation for these? Long story short, you can see at the characteristics that we looked at. Yes, we can study the same population in terms of disease. Yes, we can get the treatments. But as soon as we start introducing these eligibility criteria, these characteristics of the patients, we start to lose information. And ultimately, we found that none of these trials could be conducted using real-world data. All right, so that's one use. Then we said, okay, the other use is new indications. A drug gets approved, and then there's interest in using it for a different disease, and then getting FDA approval for the different disease. Well, what if we use real-world evidence and target trial emulation there? So we identified the RCTs for these studies, looked at their characteristics, did the same analysis, and found basically the same thing. Very hard to study these populations. The inclusion-exclusion criteria are very narrow. The patient characteristics, you can see here, we have a 33% clinical indication. That means that these diseases are so narrowly defined and so specific that it's hard to actually accurately capture that using structured information in our data set. So we had one trial here that we think could be conducted. And then we did it in another setting. We thought, okay, what disease area is it difficult to conduct randomized controlled trials? Well, substance use disorder, there are many ethical issues. There are challenges with recruitment and retention. We identified all of the RCTs on clinicaltrials.gov for substance use disorder and found that these would also not really be feasible to emulate following the true target trial emulation framework. So where does this leave us? These are my conclusions that I'm not forcing on anyone. Emulated trials may serve as a complement to randomized controlled trials. So what we're seeing here is even though in some cases we're getting directionality that may be the same, they're not here to replace randomized controlled trials, in my opinion. This is not what their use is. And this is because there's notable challenges. Until we have improved databases, improved capturing of information and structuring of information, we're going to be missing too much to really conduct analyses that adjust for all of the outcomes and confounders of interest. And this is kind of a theme that's come up in other talks as well, a triangulation of sources. Think about this as another way to look at information. Maybe this is a way to look at safety if we can't conduct another trial. Maybe this is a way to look at something that is not actually being compared and never will be compared in an RCT. But to replace the traditional RCT that's leading to approval or informing approval, this seems challenging. So I'd like to acknowledge my funders, US Food and Drug Administration, NIH, National Institute of Health, Arnold Ventures, Johnson & Johnson for data sharing purposes, and the teams that I've worked with both at Emory and my former institution at Yale. And once again, thank you so much for listening and welcome any questions.